Hello everyone, I'm Mr. Furlong, and today we're going to take a look at what's inside of cells. Oftentimes cities are being used as an analogy for cells because in a city there's lots of things that are going on in order for that city to be properly run. Well, the same is true about cells as well. There's all kinds of different structures inside of the cells all working together in order for the cell to do its job. In this video we're going to take a look at what some of these structures are. They are otherwise known as organelles. So let's start going through these. One organelle is a ribosome. Now, ribosomes are made from ribosomal RNA or rRNA, and this is where proteins are going to be made. So this is the site of protein synthesis. We find ribosomes in all forms of life. So everything from bacteria to fungi to plants and animals, we all have ribosomes because we all have to make proteins. One interesting thing about these organelles is that they are not membrane bound. So prokaryotic cells like bacteria do not have membrane bound organelles, but they do have ribosomes. Another organelle is the endoplasmic reticulum, otherwise known as the ER. And the job of the ER is to transport materials through the cytoplasm. These materials are typically going to be proteins. And we have two different types of endoplasmic reticulum, smooth ER and rough ER. And if you take a close look at the rough ER, that has ribosomes attached to it, whereas the smooth ER does not. That's really the big difference between these two. Now the job of the rough ER, of course because it has those ribosomes, is the ribosomes is the place where proteins are made, and those proteins are going to then go into the ER because these proteins are going to be used outside of the cell. So by being made on the attached ribosomes, these proteins aren't going to get mixed in with the cytoplasm. Now there are other ribosomes out in the cytoplasm that are going to be used for proteins that are needed inside of that particular cell. The smooth ER has the same function as the rough ER, just lacks the ribosomes. Now these proteins will travel from the rough ER into the smooth ER and then make their way over to the Golgi complex. The Golgi complex is a series of flattened membrane sacs. So what we can see on this particular picture is a cross-section of these membrane sacs. And if you look closely, we see these vesicles that are either coming off of it or going towards it. Some of these vesicles that are going to the Golgi complex were made from the smooth ER. And once in the Golgi complex, there's going to be some more folding of the proteins, and then it's going to package them up to be exported out of the cell. So they're going to end up in another one of these vesicles and make their way over to the cell membrane. The next organelle is the mitochondria. The mitochondria has two membranes. It has an outer membrane and an inner membrane. The inner membrane is called the cristae. That inner membrane is also a series of folds. So whenever we see folds, it's increasing the surface area for something to happen. Specifically, what's happening in the mitochondria are the second and third part of cell respiration. So the Krebs cycle is taking place in the matrix of the mitochondria. That's the liquid part of the mitochondria. And electron transport will take place on the cristae, which is why that inner membrane is folded. It gives more places for electron transport to take place so we can extract energy faster from our food that we're eating. Another organelle are the lysosomes. So these are simply vesicles that contain very powerful digestive enzymes. And lysosomes can do a couple of things. One, they can destroy foreign substances that might get in the cell. So if there's a bacterium or a virus that gets inside the cell, the lysosome can go in and destroy it. However, the lysosomes are also able to destroy the cell itself in the process of apoptosis. Our next organelle is the vacuole. Now vacuoles can be found in both plant and animal cells. We're going to take a look at it in the plant cell. And they do lots of different things. So they can store macromolecules. They can also store waste products in the plant cell. But there's a few other things that it does for the plant as well. So for instance, you may have noticed that sometimes on a hot summer day, your plants outside start to wilt because they're losing water. So the vacuoles can store excess water, which can add pressure inside of the cell. This is what we call Turgor pressure. That sort of bulges out the cell, and it makes the plant look a little bit livelier. But when there's not enough water, the cell doesn't have as much turgor pressure and it begins to wilt. And you can actually see that in a plant with enough water or a plant with not enough water. Our last organelle is the chloroplast. So these are found in both plants as well as in photosynthetic algae. They also have two membranes like the mitochondria. There's an outer membrane and an inner membrane. Inside of the chloroplast there are these disc-like structures called thylakoids. Now these thylakoids are arranged in stacks called a grana. What happens in the chloroplast, specifically in the thylakoids, is where the light reactions of photosynthesis takes place. It's in the stroma, or the liquid portion of the chloroplast, where we have carbon fixation, or what we might know as the Calvin cycle, that takes place. So when we look at some of these similarities, think about this. The light reactions are actually very similar to electron transport that happens in the mitochondria. They both happen on membranes. The Calvin cycle can be a lot like the Krebs cycle, which both take place in the liquid part, the matrix in the mitochondria, and here the stroma in the chloroplast. So there's a quick review on these organelles, and I'll see you in class.